Hello everyone, good morning. Okay, on behalf of CNRO Hall of Science and Education Technology Unit, I welcome you all for today's special lecture on the interactive lecture program in biology. The title, The Science for All, Young and Old, Rich and Poor, Amateur and Professional, Students and Teacher. Okay, we are having almost 1 hour lecture and we have interactive session around some 10 to 15 minutes. You can interact with a scientist and the online participants also. You can comment your questions in the comment box, or you will unmute, and that time you can ask the questions and you can interact. In between also you can have, if you have any questions, the audience, and you can ask the questions. Okay, and one more thing, the same thing, and we urge you to actively participate in the interactive session, the question and answer session. Okay, you could ask your questions, like I said. Fine, and thank you all for your cooperation. Now I request Professor Shiba Vasu to introduce our the guest. Thank you, madam. And Professor Shiba Vasu is the neuroscientist in JNCASR, and she's also one of the eminent scientists in the world.
Good morning, everyone. It's an honor and privilege to introduce Professor Raghavendra Gadagkar to you today. I'm sure several of you have already Googled him up and probably checked online at his profile. So I will not read out his CV. It's a very, very long list, and I will probably not do justice at all. But I will just say a few things in the manner of introduction. He's currently the Year of Science Professor at the Center for Ecological Sciences at the Indian Institute of Science, Bengaluru. He is also, we are lucky at JNCSR that he is also an honorary professor of JNC. In addition to being an honorary professor here, he's also an honorary professor of the IISER-Mohali. Some of you may have heard of this institute called the Indian Institute of Science, Education and Research, the Mohali campus. Also, he's a non-resident honorary professor at the Institute for Advanced Study in Berlin. Over the years, he chose to study a very, very fascinating question. That is, how does sociality evolve? How do some organisms have evolved to become so intrinsically dependent on one another as a species? He, in fact, chose a model organism that many of us have seen in our homes or in our neighborhoods, but he actually went on to study this model for so many decades that it has almost become a textbook example to understand the evolutionary processes that might shape sociality. Over the years, he has published about 300 research papers and articles and several books as well. In recognition of his scientific contribution, he has been awarded several prizes and awards, some of the significant ones being the Shanti Swarup Bhatnagar and the B.M. Birla Science Prize from Indian institutions. He is also awarded the Third World Academy of Science Award, which is a very prestigious international award. He has also received the Cross of Order of Merit of the Federal Republic of Germany. I think the long list I do not need to read. I'm sure you will find out. But for me, at least, I think some of the reasons that make Professor Gadagkar a very special scientist is his enthusiasm and ability to communicate some very profound concepts with such clarity and simplicity that you go back carrying that information without too much effort. He manages to convey this information with such ease and simplicity that you don't need to go and read much at all. His classes that I have had the privilege to attend have been so much fun and also very, very enriching. He has actually this series of articles that he writes that I encourage many of the students, even in my institute, to read or to go look up. I would encourage you also to look at it. It's on the wire. It's called More Fun Than Fun. Please note that down and make sure to go and look it up. I'm sure you will enjoy today's talk. I will not stand between you and our today's speaker, Professor Gadagkar. Please, we welcome you.
Good morning, everyone. Good morning, sir. Can you hear me clearly at the back there? Yes, sir. Wonderful. Well, it's a great pleasure for me to be here this morning and to speak to you. I'm going to try and remove some misconceptions of science which are in the minds of most people. And I want to explain to you that science is something not necessarily done by famous people. It can be done by you. That is the message I'm going to give you today. And let me begin by reminding you what is science. Science should be a curiosity-driven, playful exploration of the natural world. Unfortunately, modern science has become an expensive, almost industrial activity, heavily dependent on sophisticated laboratories and expensive equipment. Now, I say unfortunately because this has some very negative consequences. I will list two kinds of consequences. There are local consequences for us. This has had the effect of making most people, and especially young students, feel that doing science is beyond their reach. And all they can do is to learn what other better endowed people have done. You're all given textbooks and you're supposed to read and mug up things, and you're not supposed to produce knowledge. You're supposed to consume knowledge. This is the misconception. In my opinion, this is the most serious problem facing science education, especially in India. We all think, students think that they have to consume knowledge, teachers produce knowledge, actually teachers transmit knowledge, scientists produce knowledge, and Indian scientists think that Western scientists produce knowledge and we consume them. This has to be removed. This also has global consequences. At a global level, this tends to make developing countries like India mere consumers rather than producers of knowledge. And yet, many major new discoveries in science still come from unsophisticated, playful exploration. I want to emphasize playful exploration. That is really the crux of science. Not what result you will get, what award you will get, what disease you will cure, all that will come later. It will come on its own. Even if it doesn't come, the pleasure of playful exploration should be sufficient. That is the message I want to give you. I want to say a word about science and technology. We should not confuse between science and technology. A high school student or a retired school teacher may or may not be able to contribute to the development of new technology. Sometimes they are able to, but mostly they are not able to contribute to the development of new technology. But they can surely contribute to the development of new knowledge. That's not a problem. But we don't realize that, and we don't explore in that fashion. I want you to pause and reflect on this cartoon, which I like very much. The left panel is called Big Science, and the right panel is called Little Science. Now see what Big Science needs. Paper, pencil, and if possible, a chair. You recognize the person who is depicted on that chair? Okay. And you can have sophisticated laboratories, big factories, etc. Strictly speaking, that is little science. The big science comes from thinking. Today what I will do is, I've told you all of this, and why should you believe me? I want to convince you that this is true. And to convince you, I am going to use my own field of natural history and animal behavior. I study natural history and animal behavior, and I'm going to use my field, and I will illustrate how interesting experiments can be performed by almost anyone. The only requirements are you should be curious and you should be committed, and without necessarily having access to sophisticated laboratories. You see on this picture, on this slide, three pictures. On the left side is an ant colony. In the middle is a beehive. On the right side is a wasp colony. Now, it was already mentioned that I study a species of wasp. So this is the species of wasp that I study. It's called Rhopalidia marginata. I'm going to take 3 experiments today, one using bees, one using wasps, and one using ants. Not this particular wasp, but another kind of wasp. And I will take you through these 3 experiments and convince you that what you need to do science, what you need to discover new knowledge is not sophisticated laboratories, is not money, is not funding, is not a lab coat, but interest, curiosity, and passion. I'll take you through these experiments slowly and try to make you understand how these experiments were done and how hundreds of similar such experiments can be done by all of you. Now, I use the word animal behavior. This also has a technical name. The technical name of the study of animal behavior is called ethology. So if you just take any dictionary, you will find that ethology is a scientific and objective study of animal behavior, usually with a focus on behavior under natural conditions. But sometimes it's also useful to work in the laboratory, and I will show you how. I'll give you examples of both working in nature and working in the laboratory and show you how both are useful. But you have to begin with nature. I want to give you a more sophisticated definition of ethology. And here is a picture of one of my scientific heroes. His name is Sir Peter Medawar. And along with his wife, he wrote a book, which is a very interesting title. The main title of the book is Aristotle to Zoos. And the subtitle is A Philosophical Dictionary of Biology. You all know dictionaries. You know the word philosophy. So you can imagine what a philosophical dictionary is. And since it's a dictionary and has entries alphabetically, they have conveniently called it Aristotle to Zoos. The first entry in the book is about Aristotle, and the last entry of the book is about zoos. This is the only dictionary I know which you can actually read from cover to cover. And you should read from cover to cover. It's so delightful. In this book, they talk about ethology. So they're not just, this is not an ordinary dictionary. It's a philosophical dictionary. So this I gave you from the Merriam-Webster's dictionary, what is a dictionary definition of ethology. This is the philosophical dictionary of ethology. The word ethology is not merely an alternate designation for the science of behavior. It's not just that. It is a term that stands for a genuine revolution in biological thought. Ethology is rooted in observation of animal behavior, an activity that only simpletons think simple. This is the philosophical touch that they are giving. And they go on to say that observation is a difficult and sophisticated process, calling upon all the intellectual virtues: attention, patience, heightened awareness, caution in coming to conclusions, courage in framing expectations. This is what you need. Not money, laboratory, instruments. That's not what you need. What you need are attention, patience, heightened awareness, caution in coming to conclusions, and courage in framing expectations. And I will illustrate these phenomena in the 3 experiments that I have chosen to talk to you about today. I will tell you towards the end of the lecture in more detail, but let me just tell you that I have written a book about such experiments. And the book has 15 experiments. From that book, I have chosen 3 to discuss with you in the short time that is available. I will give you the more information about the book, but you can go and read about 15 such experiments, which any one of you can do, and you will find much more detailed information in the book than I can give you today in the talk. Ethology really was born with Charles Darwin. People know Charles Darwin as the father of the theory of evolution, but he was actually the father also of ethology. He wrote a wonderful book called The Expression of the Emotions in Man and Animals. This is the kind of language they used in those days. We say behavior, he used the word emotion. It's a very detailed book, but this is where ethology was born. And ethology has matured. It has matured so much that in 1973, 3 people were ethologists were given the Nobel Prize, which is very unusual. Because you have Nobel Prize in physics, you have in chemistry, You don't have a Nobel Prize in biology. It's called physiology or medicine. But the committee which decides on who should get the Nobel Prize was so convinced that these ethologists had contributed so much towards physiology and possibly towards medicine that they actually awarded them the Nobel Prize. On the left side you see Carl von Frisch, who is well known for discovering the honeybee dance language. In the middle panel you have Konrad Lorenz, who was famous for discovering imprinting in birds. And on the right side you have Nico Tinbergen, whose experiments I will actually talk about. So let us begin. So the first experiment I have taken from Nico Tinbergen, who was the third person on your slide. Now, Nico Tinbergen was a Dutch scientist, but he spent most of his time in Oxford, and he of course got many awards including the Nobel Prize. He illustrates what is meant by curiosity about things happening in nature. He would love to go for outdoor walks for pleasure, and he would see things and he would become curious about them. Just by this trying to satisfy this kind of curiosity, he discovered many things, many experiments, and obviously they were important because he even got the Nobel Prize, right? Now I will take just one example of curiosity driven research. Where he was walking, there was a little in the woods, and he found many individuals of this wasp. This is not a social wasp, this is a solitary wasp. And what solitary wasps do is they dig a hole in the ground, then they hunt for a suitable prey item. Then they will actually sting that prey item and paralyze it, because the venom in their sting is a neurotoxin. So when that venom gets into the victim, that victim does not die, but it's only paralyzed, which means it cannot move away, but it is alive. So then it will put that prey item in the hole, lay an egg, seal it and go away. Many days or weeks later, the egg will hatch a little larva, and it will be sitting on a pile of food, which is the paralyzed prey. And because it is paralyzed, it has not run away, it has not walked away, it is very there. And because it is not dead, it is alive and fresh to eat. The larva then consumes this prey item and becomes a pupa in the same hole, and then comes out an adult wasp. And the cycle repeats. Of course, both male and female wasps are produced. They mate, the males die, and the females lay eggs before they die, and this process continues. And when Tinbergen was walking around, he found a very large number of these female wasps, very busy. What were they busy with? They are digging holes in the ground and going out and bringing prey items. In this particular case, their prey item happens to be the honeybee. In fact, that they hunt bees, and so they are commonly called as bee wolves. So this is a wolf that hunts bees. So they will go and catch hold of honeybee, sting it, insert it, but they need many honeybees because the larva needs a lot of food. So they go back, find another honeybee, sting it, put it back here, and they are very busy doing this. Now you can admire this, but you have to go beyond admiration. You have to be curious, you have to ask questions. So Tinbergen said, you know, what really impresses me most about this wasp is there are so many wasps, so many holes, they are all going back and forth. How does a wasp know which is my hole? They don't seem to make mistakes, they are not hesitant, they rapidly go back, they are completely confident. And all these holes, I cannot tell them apart, they all look the same to me. How does a wasp know which is my hole? But they must know, because I don't want to go and put food in somebody else's nest to feed somebody else's larva, right? A wasp should go to its own hole. How does it do this? This is the crucial thing about science. You must ask questions, you must be curious, you must not be satisfied. When poets look at nature, they are satisfied. They sing songs in admiration. But the real poet will also ask a question. And if you ask a question, and if you find an answer to that question, you can write even a better poem. So you must ask questions. You must never just be content with what you see. How does this happen? Why does this happen? When does this happen? You just walk anywhere, you must ask questions. You must get into the habit of asking questions. Now, most of the questions you ask cannot easily be answered, but just asking the question is a pleasure, and occasionally you can try to answer that question. That is how science is born. So but it is not enough to ask questions. You must make some educated guesses and see which guess is correct. So Tinbergen made 2 educated guesses. He said one possibility is that they use their eyes, visual cues. So they know which is my hole because they have studied very carefully that the surrounding of my hole or nest is really different from that one. Seems very difficult for us, but maybe the wasp can do it, who knows. To us they all look alike, but we are huge. Their wasps are small, they go close by and they see. Maybe there is sufficient variation in the landmarks around each nest that the wasp studies this and is able to remember it and therefore make no mistake. So possibility, this is a guess. Technically we call this a hypothesis. But it's alright if you use the word guess, that's fine. But it is not proven, this is just a guess. It may be true, it may not be true. So this is one guess. It's very nice often to have 2 guesses at least, so that you can compete between them and you can test between them. He said, okay, I have a second guess, maybe they don't use their eyes, maybe they smell, maybe they use their nose. Now wasps don't have noses like we do, but they have antennae, and their antennae function as their noses. They have receptors in the antennae, they are very, very sensitive to smell. So maybe each nest smells differently, and by smelling, by sniffing out their nest, they know which is mine. These are 2 possibilities. Now, how to know which is right? Just let us do a simple experiment. The impression is that experiments have to be complicated, experiments have to be precise, experiments have to be sophisticated. Not true at all. The best experiments are called quick and dirty, simple experiments. So Tinbergen said, let me see whether they use visual cues. Can I provide evidence that they use visual cues? Which means they are memorizing the landmarks around each nest. How do you test this possibility? Now I will tell you what he did, and then you must understand how you should do things in a similar situation. He did not go and take detailed photographs of the nest and do a computer analysis of how the surrounding is different from each nest, because probably the wasp doesn't do that. He said, let me change the surroundings and see whether I can confuse the wasps. How do you change? It's not easy because you don't really see anything special. So he said, let me exaggerate the visual marks first, and then it's easy for me to change. How do I do that? He looked around, he found lots of dried pine cones lying on the ground. He picked up these pine cones and he arranged them around the nest. So now you can see this is a much more exaggerated visual mark, which the wasp should definitely be able to see. Even Tinbergen was able to see, and he said, now I will test. He did a very simple experiment. He placed these pine cones around the nest and allowed the wasp to fly back and forth several times. Now the wasp should have learned that the visual marks around my nest have become exaggerated. Now they are even easier. Some good soul has helped me to make it even easier for me to find my nest. Very good. So I will learn these, I will go and come back. Now Tinbergen said, after several trials, he said, maybe now the wasp has learned. Now let me put the wasp to a test. You know, you are taught in class and then you have an exam, right? So he said, let me give an exam to the wasp. And he did something very simple but very interesting, very clever. Next to the nest, he dug another hole by himself, but this is a false nest. There is no larva, there is no food, there is no nothing. This is something that, this is a Tinbergen's nest. And technically we call these such things as sham nests. That means they are fake nests. And then what he did, when the wasp was away, to do good science you must also be a little bit mischievous. when we always scold students, don't be mischievous, but we also kill their curiosity by doing that. You have to be a little mischievous. He said, let me fool this wasp. When the wasp was away, he removed the pine cones from the real nest and placed it around the sham nest. Now the wasp should get fooled. It should, if it is using visual cues, it should get fooled and it should go here rather than its real nest. Can I fool the wasp? If I can fool the wasp, then I have some proof that it is using visual cues. If I cannot fool the wasp in spite of this exaggerated landscape which has been moved, then it is probably not using vision. Do you get the point? So let us put the wasp to a test. So after he changed it, he waited for the wasp to come back. All he had to do was to observe whether the wasp will go here or go here. And he found, to his great surprise and to his great delight, that the wasp was completely fooled. Every single time it went to the wrong nest. Never did it go to the real nest, which it had been doing all these days. So clearly it is using visual cues. Now one of the important things for a scientist, never be satisfied. Every answer you get, every discovery you make is tentative. It may be wrong. You have to go further. So never be satisfied. He was not satisfied with this. He said, now if this is true, if my guess that they are using visual cues is true, I must put the wasp to one more test. Now I should, when the wasp is away, I should take the pine cones, put back on the real nest and then see where it goes. Now the same wasp should go to the real nest, not to the wrong nest. So this is the second experiment that is essential. Why is it essential? The wasp, yes, went to the wrong nest, but maybe the wrong nest was very nice. Maybe it said, oh, this is a better nest, let me go here. Not because it got confused. It actually likes it. So you have to worry that the animals are always smarter than us. So it's not going to the new nest because it likes the new nest, but because it got fooled. And to prove that, you put back the cones on the real nest and now ask what to do. Every single time the wasp went to the real nest, giving you much more confidence that they are using visual cues. Don't be satisfied, because maybe they are also using olfactory cues as well as visual cues. This only proves that they are using some visual cues. How do you know they are not using smell? They may also be using smell. So he did another very simple experiment. This time, not only he put pine cones around the original nest, he took two small pieces of cardboard. I want to emphasize that you don't write to Government of India saying, I want 20 lakh rupees, I am doing research and order something from America. You take a piece of cardboard, dipped it in some scent, any scent you like at home, okay? And he puts two scented cardboards here. So what is he doing now? He is exaggerating both the possible visual cues as well as the possible olfactory cues, smell cues. Both are there now. Now what does he do after so wasp goes back and forth, it learns everything. Now during the exam for the wasp, what does he do? Very clever, notice this. He moves the pine cones to the wrong nest, retains the scented cardboard on the old nest. Because if it is smell, they should go here, if it is vision, they should go here. Now there is a competition between these two. That is why it is good to have two hypotheses, two guesses. But notice that he also put two pieces of cardboard on the right side, which are not scented. Can you guess why? Sorry? Yes, because after all, you could take the scented cardboard and treat it as a visual object. So then the wasp will get confused because some visual objects are here, some are there. So unscented cardboard on the right side, and now ask where will the wasp go? Every single time it went to the wrong nest, ignored the scented cardboard and went to the pine cones. Put everything back, every single time it goes to the real nest. So now you have much more confidence that using visual cues and not olfactory cues. Never be satisfied. In fact, you should never be satisfied. As long as you live, you should not be satisfied because there may be something further. Science is an endless process. Science is a process. It's a journey. It's not a destination. The impression we always create is that, I have done this, I have got this prize, now I have my end of it. There is no end for science. Science is a journey. There is no end station. You must learn to enjoy the journey. It's not saying, I have to take a train, I have to reach Delhi. You don't have to reach anywhere. Just keep, it's nice to have you can have a circular train. You can keep going around and round, and it's a journey that you should enjoy. So Tinbergen did not stop here. He said, he used other experiments, which I won't go into the details, and he did many new experiments. I'll just give you the final result of these experiments. He showed that cones, so he said, maybe the cones also have some smell, and maybe because of the cones, they are still using smell. So I want to get rid of the smell of the cones. So he dipped the cones in alcohol overnight, dried them, and used cones, removing the smell, and still the wasps got confused. De-scented cones also work just as well as scented, naturally scented cones. So further confirmation. Wasp with, now we know, I told you the nose of the wasp is in its antenna. So cut off the antenna. Wasps with amputated antenna also learnt visual landmarks. That means they didn't even need their antenna to go correctly to their nest. Each of these is an experiment. I am just giving you a few examples. The wasp could not be trained on scent alone. Now he said, okay, let me remove all visual marks, let me give them only scent, and they never learned. So you see, he is amassing proof in favor of the vision hypothesis and against the olfactory hypothesis. The visual landmarks were recognized from up to 100 meters away. Up to 100 meters away they were able to, and he is very cautious in saying, I don't know what they use before they come to 100 meters. I have no idea. I can only talk about what they do when they come close by. So he made many discoveries. What did he need? Curiosity, playfulness, not sitting in the classroom but roaming around in the forest, in the woods. Discoveries can be made anywhere. You don't have to sit and read necessarily, but you must think. And you must not be satisfied. You must be curious, you must be passionate, but never satisfied. The worst enemy for a scientist is satisfaction. Even if you get the Nobel Prize, you should not be satisfied, because there is no end to science. Let me now illustrate a second experiment. This experiment was done by the other Nobel laureate I told you about, Carl von Frisch, who discovered the dance language of honeybees. I won't talk about the dance language, but I'll talk about an experiment, a very, very important discovery that Carl von Frisch made even before he discovered the dance language. And here again I'll illustrate all these principles: curiosity, passion, not being satisfied, cleverness in framing expectations. Okay. This is Carl von Frisch. He was an Austrian zoologist, and he worked with many organisms, but he is best known for his work on honeybees. But he also did very important work on other organisms, especially fishes. Now what you see here is Carl von Frisch sitting. He has put a little stool in the garden, and he has kept a little bottle with sugar solution, and bees have come there. He learned that you can make the bees come to you rather than you go to them. See, bees collect nectar and pollen. Okay, I don't know whether I have slides to show that. Okay, so this is, for example, a picture of a honeybee call. You have all seen this. A large number of bees are clinging to a nest. The nest is made from wax, and they make it themselves. And inside this there are hexagonal cells in which their legs. Now in each nest you have a single large queen, a small number of male bees which are called drones, and thousands of worker bees. The worker bees actually do all the work. They build the nest, they clean the nest, they repair the nest, they guard the nest, they go out and bring food. Their food consists of two items: pollen grains and nectar, both collected from flowers. Bring it back, store it, process it, mix the two, and feed the larvae. In a honeybee colony there may be 10,000, 20,000, 30,000 hungry larvae waiting to be fed, and that we fed several hours every day. And that's what these workers do. Here is a bee on a flight visiting a flower, and what you see here actually are bundles of pollen grains. So when the bee goes to a flower, it goes there to collect nectar. It is not collecting pollen actively, but the pollen rubs off on its body because the bee is a very hairy individual, lots of hairs on its body, and the pollen gets stuck. And on the flight back, it actually uses its legs to pack all of this pollen on its hind legs, little structures in the hind legs, which are called pollen baskets. And whatever nectar it has taken, it has stored in a temporary place called the crop, where it doesn't get digested, it doesn't drink it, it just stores it, goes home and deposits both the pollen and the nectar at home. Von Frisch discovered something very nice. He said bees don't collect food for themselves, they collect for the colony, because all these larvae and the bees at home have to be fed. So one consequence of this is bees are forever hungry. If you are collecting for yourself, very soon you will say, enough, now I don't need anything till tomorrow or till today evening. But the bees are collecting, they want kilograms of honey, kilograms of nectar, so they are always hungry. This hunger, this perpetual hunger of bees can be exploited in a very useful way. So what he did was he went close to a beehive and he kept a bottle of sugar solution. And the bees accidentally discovered this because it was very close by. They were going around and said, ah, there is some sugar here. I don't have to go to that tree so far. They collected that sugar, went home, deposited it, performed a dance and told other bees, come, there is sugar very nearby, let's go. So lots of bees will come and start taking it. After some time, you have to be, as I told you already, you have to be a little mischievous. He said, now I want to fool the bees. So he moved that table with the sugar a few meters away. So the bees came and they didn't find it anymore. They were very agitated. The bees also don't give up. They looked around and very quickly found the new one, the displaced one, because it was not very far away. Very good. So back and forth, back and forth, back and forth. After some time he moved it some more. No problem. They discovered it soon, back and forth. He showed that he can move this up to 10 kilometers and the bees will come. You can take it to the third floor, they will come. You can take it behind the tree, they will come. In fact, my students train the bees to come to the lab, go to the third floor, enter through the window, come and sit on the microscope stage where we want to observe them. Because you train them, they will do it. They are hungry. All they need is food. You can make them do anything. Using this, not just Carl von Frisch, but thousands of scientists have been able to do wonderful experiments because the bees are so cooperative. They will do whatever you want. All you have to do is give them some sugar. There's one little problem. There are also trees competing with you. Trees are also giving them sugar. So what you have to do is you have to make your sugar more concentrated than the nectar in the surrounding area. That's all you have to do. In fact, if the trees are not giving good nectar, then yours is so much better that too many bees will come. You don't want so many bees. No problem. Reduce the concentration of sugar. So by adjusting the concentration of sugar, you can make 1 bee come, 2 bees come, 5 bees come, whatever you like you can do. They are wonderful to work with. But you are not satisfied with the wonder. You must ask questions. And you have to train yourself to ask questions. Sometimes we don't even realize that this is a question to be asked. So we have to practice. Why do we have 5 fingers? Why not 6? This is a question. You may not find the answer easily, but don't be satisfied with any information given to you. Try to question why. Now how do you begin to answer this? It's not easy. You can begin by saying, okay, I have 5 fingers. Does everybody have 5 fingers? Well, most human beings have 5 fingers. Do all animals have 5 fingers? The answer is no. Some have less. What is the difference? How do they use their fingers? That's how you begin. But everything is a question. So von Frisch asked a very simple question. He said, flowers are beautifully colored. Why are they so brightly colored? Yes, but it can't be for satisfying us, right? Why not for satisfying us? It may be, make humans happy, make gods of humans happy. Why not? What is the scientific answer? Suppose I say, I believe that flowers are brightly colored in order to make humans happy, in order to make humans pray, offer their gods colorful flowers and get good blessings from the gods. How do you disprove this? How do you know this is not true? This cannot be true because bees evolved 50, 60 million years ago. There were no human beings at that time. Flowers evolved 50 to 60 million years ago or even more. There were no human beings at that time and they had brightly colored flowers. So they could not have done this to satisfy humans. Humans are a very recent addition to the biota of this planet. So trees evolved long before humans. So it could not possibly be for humans. So it should be to attract bees. But Fawnfish immediately knew that this answer probably is wrong. Why is it wrong? Because if it is true, then bees must have color vision. They must be able to distinguish between different colors. And in Fawnfish's time, it was very clear that bees cannot possibly have color vision. They are so small, they are so tiny, just an insect. How can they have? Many higher animals, many mammals, the famous bull which you enrage with a red cloth is color blind. So how it's not possible? So it was widely believed that bees cannot possibly have color vision. Therefore, this answer must be wrong. Worse than that, an eminent ophthalmologist called von Hess, who had written an authoritative book on vision science and had been knighted for his work. Know what he was knighted? Sir C.V. Raman. Okay. He had conducted on the basis, but he had some very sloppy experiments under unnatural conditions. Remember, natural conditions where you have to begin with. He had conducted sloppy experiments under unnatural conditions and concluded that all invertebrates and even fish were color blind. This was the authoritative knowledge at that time. Fawnfish was not impressed and decided to conduct his own experiments. This is the kind of arrogance that you need in science. Your teacher says something, why should I believe? Now forget teacher, this is the most famous person at that time. I don't care. Because to me, if this is true, if bees are colorblind, why are flowers colored? I have a problem. So I will do my own experiments. Fawnfish was 20 years old and this gentleman was some 60 years old and he was the most famous person at that time. He did very simple experiments. In fact, the experiments are so simple that these are repeated by students in my lab all the time. Attached to my institute, there is a Kendriya Vidyalaya, and 10th standard students are supposed to show that they can do some experiments. So they always come to my lab and said, sir, I want to do some experiments. And I make them repeat Carl Fawnfish's experiment. The experiment is very simple. We take a cardboard box and we put some sugar solution inside the cardboard box. And we have two holes in the cardboard box from where the bees can enter to drink the sugar solution. Now, we put cardboards with different colors around these holes. Just like tin burgeon had two nests, pine cones one, not in the other. So here is a green cardboard and a blue cardboard. Nothing very fancy. This is something you can buy for 1 rupee. And what we do is we keep one of the holes closed and allow the bees to go through the other one and learn the color. And we then give the exam to the bees, very similar to tin burgeon's experiment. Now this is what an experiment, the data will look like. This is not data from Fawnfish. This is data from students who come to me from Kendriya Vidyalaya. Now I want to explain to you what this means. On top it's written training. So this is how you train the bees. Then there is a test. We will come to the test later on. What is written there is blue plus, green minus. What does that mean? Blue is open, green is closed. That's all it means. So during training, the blue was closed and the green was open. Okay. And during test, what happened? If bees have color vision, they should go to the blue and they should avoid the green. So you can see that 104 of them went to blue, only 2 went to green. I think that's a good percentage of marks that the bees have got, right? Nevertheless, you must ask yourself, Why did 2 bees go to the green? Are they stupid? Think about it. But you must always worry about these things. But for the moment we will say, if student gets 70% marks, we can ask why didn't he get remaining 30%? What is it that he or she does not know? You can ask that. But for the moment we will pass this bees and we will say yes. But you should not be satisfied. Von Frisch said, I cannot conclude that the bees have color vision because I put the blue on left side, for example. In this case it was right side. Maybe the bees like the right side. They don't care about color. I like the hole on the right side, so I go there. I don't like the left side. So how to prove that? How to disprove that? Very simple. You put blue on both sides now during the exam. If they like right, they should only go to the right. But more or less equal numbers went to both sides, which means they are not learning the side. They are learning the color. But you should not be satisfied. What you should do, you should always, there's a way to say this. It's called playing the devil's advocate. That means always saying the wrong things. You must either you have a friend who doesn't believe you, or you yourself train not to believe yourself. Von Frisch said, no, I don't believe this. Maybe bees just love blue. They don't like green. Possible. So how to disprove that? Repeat the experiment, blue minus green plus, and test, and of course they all went to the green. So it's not, they have no great dislike for green. 100 out of 103 went to the green, and again it's not side because I have put green on both sides, they go equally. So now you have more confidence. Do you have adequate confidence? No. This does not prove that bees have color vision. You are all too young to have experienced a black and white television, but if you look at black and white TV or black and white photographs, different colors will appear as different shades of grey, and you can tell them apart. You don't have to have color vision for that. So maybe the bees are just distinguishing different shades of grey. Maybe they don't have color vision. So you should always question your own finding. You must always be the enemy of your own self. And you must say, no, what I have done must be wrong. My conclusion must be wrong. Keep on doing this, and many times you will try to disprove yourself, and if you don't succeed, then you are probably right. So how did he check the possibility that they are distinguishing blue and green? Because they appear as two shades of grey. Again, very simple. He went to the market and he went to a shop where they sell different kinds of paper, and he told the shopkeeper, give me a small piece of paper of all shades of grey that you have, and I am going to compete the blue color with all possible shades of grey. At least one of them should be confusing. If none of them are confusing, then I have little more confidence that the bees have color vision. This is what he did. He used different shades of grey. Do you understand the logic of his argument? He says, if they are distinguishing blue because it appears a certain shade of grey, then at least one shade of grey should be confused with the blue. They should not be able to distinguish between blue and a particular shade of grey. But all shades of grey were rejected by the bees, and they went to the blue. The story had a happy ending, but before the happy ending, there was a problem. Shockingly, the great ophthalmologist C. von Hess refused to accept von Frisch's results. He insisted that it was possible to demonstrate that the older claims of somebody called Labocque, as well as the recent ones of von Frisch, according to which bees can be trained to certain colors, are wrong altogether. The famous man wrote. And not a single fact is known that could even make plausible the notion of color sense in bees. Through my investigations, this assumption is terminally refuted. After all, he is Sir von Hess, sir. He can say that. Von Frisch was 20 years old. Did he buckle down? No. He said, I stick to my thing and I am right. But he was a human being after all. It was a very touching story. He wrote a letter to his mother saying, Mama, I think I have one enemy in the world. But he didn't give up. The young von Frisch stuck to his position, and eventually von Hess was forgotten and von Frisch got the Nobel Prize. More recently, we have of course much clear proof. The ultimate proof of course came from more recently in the 1960s, the physiologist Outram obtained direct evidence of trichromatic color vision in bees. By inserting microelectrodes into the single photoreceptor cells of bees and recorded electrical activity. You may have seen these kinds of things. So this is for humans. I've used Mona Lisa here to represent humans. So we have a blue, green and red. These are our three primary colors. The bees also have three primary colors, but they are not blue, green and red. They are in fact ultraviolet, blue and green. So ultraviolet light is used by bees. Bees can see in the ultraviolet, which we cannot see. In fact, if you shine ultraviolet light on flowers, they look very different. But they also have trichromatic vision. But and this of course was done using very sophisticated way of inserting a glass electrode into a single cell and recording. But what I want you to remember is that von Frisch got the Nobel Prize, not Outram. That is the difference. This was confirmation, expensive confirmation of a profound result got by very simple experiment. So the student can be von Frisch and the professor can be Outram. Who will get the Nobel Prize? The student will get the Nobel Prize. Far cry from students saying, oh, how can I do science? My professor is so famous, so rich, so learned, so experienced, and he has a big laboratory and he has lots of money. How can I compete with him? This is the problem. This is why we don't produce good scientists in this country. That has to change. But you must also remember that nothing is end. Continue. The students who came to my lab, they said, this is very nice. We were very happy repeating the experiments of a Nobel laureate, etc. But we are not satisfied. We want to do something more than what the Nobel laureate did. I said, wonderful. What do you want to do? So one batch of students said, we want to fool the bees some more. That they like the idea of fooling the bees very much. And this is very important for a scientist. He said, I want to fool the bees some more. Okay? So what do? They said, now I am going to put the beehive also into a cardboard box and make two holes there. And the bees have to, when they come to the feeder, see what it says here. Feeder is blue plus green minus. So when they come to the feeder, they have to enter through the blue hole, avoid the green. But when they go home, they have to avoid the blue and go to the green. And the feeder was here and the hive was here. That's all. So in a second, the bees will fly. And the bee should know, am I entering home or am I entering the hive, entering the feeder? And they should not make mistakes. Can the bees do that? Can they change so quickly and can they know, can they be aware of what I am doing? How will they make too many mistakes? Answer is, bees are very smart. Most of them went correctly to the blue here and to the green here. And this is within 1 second. So they learn both and they remember both and they appropriately do that. This is like a very tough exam for the bees. But the bees passed this exam. In another year, students said, we also want to fool the bees some more, but differently. So they set up an interesting experiment. They said, in the morning, only feeder, no hive. In the morning, I will keep the blue door open. In the afternoon, I will keep the green door open. Will the bees look at the watch and say, oh, it's morning, I should go to blue. It's afternoon, I should go to green. Will they do? Yes, they do. Where is their watch? You have to discover. But they know which is morning, which is. I also want you to notice that compared to the previous experiments, the number of errors in these experiments are much more. And you must think, why? Why are these errors much more? Don't come up with one answer. Come up with 2-3 answers and try to see which is more likely to be correct. Sometimes even without doing experiments, by simply quarreling amongst yourselves, you can try, that's what you should do. One student says, this is the reason, another student says another reason, and you fight amongst yourselves. Think about it. Just by thinking, you can learn a great deal. Always ask questions. Why are the errors here? The errors in the first case were very few. Here the errors are a little more. Here the errors are even more. Why? What is the reason for these errors? Do they have a bad watch? Do they not know whether they are at home? Or is there some other reason? It does not matter that we don't know the answer. It does not matter that the answer is probably unknowable. Asking the question is important. Answering is not important. In fact, the moment you answer a question, it is boring. Over. The longer it takes to answer the question, the better it is. You are reading a mystery novel. In the first chapter, if they tell you who committed the murder, you are not going to read on. And in order to make this suspense go on, what scientists do is they say, I am not satisfied with the answer. I am going to try and disprove this answer by doing another experiment. That is how science is done. Now I will come to the last experiment. This is with ants. And this is done more recently by a person who has, let me say, not yet got the Nobel Prize. This is actually a friend of mine. His name is Jean-Louis Deneuberg. He is actually a chemist. And it's very important that he's a chemist. I'm going to emphasize that. He's not a biologist. So he does not think like a biologist. He thinks like a chemist. And that was his success. I would guess that if he was a biologist, he wouldn't have done this. He wouldn't have thought about this experiment. It is because he doesn't know. You will be surprised that in science often a certain amount of ignorance is very, very useful. If you mug up all your textbooks, you will have no questions. You will have only answers. Too much knowledge is bad for science. You must have a question and you must not know the answer to that question because you are ignorant, but you must know lots of other things so that you can design an appropriate experiment. You need selective intelligence and selective knowledge. So simply reading all the books in the world, going through all entries in Wikipedia is not going to make you a great scientist. But you must know how to use your ignorance and your intelligence. You have to use your knowledge in the service of your ignorance. You need a combination of these two. He didn't know many things because he is not a biologist. And he had also an interesting question. He was interested in observing ants, just like Tinbergen was interested in observing wasps and conservation in bees, he was interested in ants. And he found that the ants are very smart. You know, they go in a line, and when they go in a line, they actually deposit some chemical, which is called a pheromone. And therefore the next ant knows where to go. So they follow each other because they are laying a trail of chemicals, which are called pheromones. And that's how they follow each other. Now he found that ants are very smart. If there are two ways to reach a place of food from their home, one is a long route and the other is short route, very quickly the ants will realize which is the short and they'll use the short. They'll avoid the long one. How do they do this? How do they know that this is long and this is short? How do they know that short is better and long is bad? How do they do this? Now a biologist would have gone in a different direction making his guesses. Being a chemist, he didn't know anything. He said the answer must all be in chemistry, not in biology. And he came up with his guess, with his hypothesis. This is the ant that he worked with, it's called the Argentinian ant, and his guess was this. He said, let us imagine that two ants leave their nest at the same time and take different paths to a food source, but both of them mark their trails with pheromone. They don't know which is long, which is short, nothing. They go randomly, some choose the long and some choose the short. But the ants that took the shorter path will return first. And this trail will now be marked with twice as much pheromone as the path taken by the second ant, which is yet to return. Because go and you come back. Now the nestmates will now be attracted to the shorter path simply because it has more pheromone. The other one, the long one, has less pheromone. So no intelligence is needed, no measurement of distance is needed, no knowledge that short is better, nothing is needed, just chemistry. The colony's efficient behavior emerges from the collective activity of individual ants following two very simple rules. Lay pheromone and follow the trail of other pheromone. That's all you need to do. You don't need to know which is long, which is short, when short is good, long is bad, nothing you need to know. Just with these two. This is how a chemist thinks. A biologist might have said, no, it can't just be true. So let's do an experiment. So this is where he had to do an experiment in the laboratory to test this. It was not easy to do it in nature. So he set up a very simple experiment. He created in the nest, in the lab, a nest of the ants and a food source, and he connected the nest and the food with glass tubes. Very simple, one short tube and one long tube. And so he can test what the wasps do. Now actually he did not do this. He made his experiment a little bit more complicated. he put two such arrangements. This is one unit, so I had one unit here, another unit here, one unit here, another unit here, and you must think about why he did the experiment, 2 units of glass tubes and not one. I won't answer that question, something for you to think about. But now you can see here that the ants are going randomly, some on this side and some on that side, but here they are using the shortest path. They are avoiding this loop as well as this loop, all are going on this. This is not the result, this is not a photograph. This is a diagrammatic illustration of the hypothesis. The hypothesis is that initially they will choose randomly and then they will all do this. This is not the result. Now you do the experiment. Here, of course, you can do the experiment, you can have food here and you can watch what the ants do. Now, since he was a chemist and with a very good background in mathematics, he also said, I will make a computer model of this. I will write a computer program which will mimic what the ants do. But first you go randomly, then you follow that around, and what the computer program should predict what should be the outcome. What is the expected outcome? That very soon they will stop avoiding the long path and most of them will go in the short path. Can the computer model show this? And what you see here is the result. So this smooth line is what the computer model showed, how it should happen, how the percentage of the winning path should increase and saturate, and this is what the ants actually did. Small errors. The computer program did not make small errors, the ants made small errors. Ask yourself why. Are they stupid or not? These are the kind you must think about this. As soon as I say, oh, so wonderful, such very, very similar, the computer program and the ants are very similar, I am very happy. Don't be happy. Ask yourself, but why? Even if there is a small difference, ask yourself why. Very often we don't ask questions because we have this wonderful phrase called stupid question. We don't, because it's such a stupid question. No question is stupid. Questions cannot be stupid. Answers can be stupid. That's easy. So don't hesitate to ask questions. So it is a teacher who has to answer a question has to be careful because answer could be stupid. Questions can never be stupid. There is no such thing as a stupid question. As long as you don't know the answer, it is a valid question. So think about why they are not exactly the same, but once again let us pass here and say it is more or less the same, very similar. So let's accept this. So this is what the ants which he studied actually did, and what he did of course is he was able to change the ratio of the tubes. In one experiment the long tube is twice as the short, in one case it is 1.5 times, in one case it is twice, one case it is thrice, one case it is 4 times. So what do you expect? The more the difference, the more rapidly they should learn, right? Do you see that? The bigger the difference between the short and long, the more rapidly they should learn, and that is what he found. Sounds very good, everything is fine? No. Jean-Louis Deneberg was a true scientist. He said, I am not satisfied. I have to be the enemy of myself. I have to do something which will potentially disprove my experiment, disprove my theory. Because, so I have to come up with something here. You have to become the enemy of yourself. That's very important. Don't be satisfied. So he said, if my theory is correct, then I should have one more experiment and I should be able to show that if I close the short tube for some time and make the ants go back and forth in the long tube, then open the short tube, then they should not be able to use short. Because there is already so much pheromone on this, no pheromone. If pheromone is important, not long or short, then the ant should not be able to do that. So he did that on his computer and he did real ants, and just as expected, the ants were not able to choose the short path anymore. By opening the short path 30 minutes later than the long path, they were not successful. They were totally lost. You can see how this gives additional confidence in your original experiment. But nothing is end. Now you must come up with something, another experiment which will potentially disprove this, because this is an endless cycle. And the more and more experiments are successful, the harder and harder it is for you to come up with an experiment which will disprove that. That is what you should be doing. Anything that you read, any experiment that you read, try to find fault with it. Try to say this is probably wrong and this is the way I can test it. Never be satisfied with anything. And that's fine, because we don't want knowledge, we want the pleasure of discovery. What do you discover? Okay, they use pheromones. So what? There is no pleasure in that. You weigh something, you know, people ask stupid questions. What is the weight of the moon? So many kilograms. So what? There is no fun in the answer. The fun is in the question. Don't ask what is the weight of the moon. Ask how do I sit on earth and estimate the weight of the moon. I don't have a balance. I can't put the moon on a balance. How will I sit here and find out, guess what the weight of the moon is? That is the kind of question you should ask, and never be satisfied. The answer is boring. The more experiments fail, the luckier you are. The more delayed the answer is, the luckier you are, because the journey continues. You don't want to reach Delhi, then you have to get off the train. That's why I said you have a circular train which keeps going round and around the country. You can keep enjoying it. The journey is more important. Now it turns out, as I said, don't worry about whether my research is useful, whether it will solve some problems, whether people will make money. Don't worry, because you don't know. When Jean-Louis Denevier, he didn't know anything. But it turned out that just as he did this experiment, in the neighboring lab, there was a computer scientist. And he was so fascinated, he said, this is of great use to me. This knowledge that you have found, that this is how the ants find the shortest path, is of great use to me. And he actually wrote a whole book saying how computer scientists can use this information. And now there is a branch of computer science called ant colony optimization. I'll just quote a little bit from him. Dorigo said, Mark Dorigo and his colleagues have adapted ant-based routing to handle internet traffic. Simulation results indicate that their technique outperforms all existing routing methods, including the current one used in protocol. Ant-based models are now being used by Southwest Airlines for efficient routing of cargo. Who would have guessed that this would have been so useful? Another ant scientist called Eric Bonobo was actually hired by France Telecom. He said, we want an ant scientist to tell us how to route telephone properly. And he says, the ultimate application of ant-based routing methods might be in the internet where traffic is painfully slow. The most powerful insight from swarm intelligence. So this is called swarm intelligence, because each ant doesn't know which is the long, which is short. Each follows two rules. Follow late pheromone, follow the trails of others. Because of that, the colony does something very intelligent, that is choose the short path. That's why we call it swarm intelligence, is that complex collective behavior can emerge from individuals following very simple rules. Possible applications of swarm intelligence may be limited only by imagination. Such things may come, but it doesn't matter if they don't come. The pleasure, Jean-Louis Denevier was as satisfied before this happened as he was. So I want to end with a few take-home messages. The process of science is as important as the product. Don't be satisfied with the answer. Somebody gives you an answer, you know, what is the specific gravity of something, what is atomic number. Always wonder, how did somebody find out? And if you think along the lines, you may be able to show that they were wrong. If you're only looking at the answer, how can you say that it's wrong? There's no way. Don't accept anything. So the process is important. It's not enough to know what the scientist found, but ask how she found it. That will give you an entry into science. That will make you a knowledge producer. If you are interested in the answer, you are a knowledge consumer. Don't be satisfied being consumers. Try to become knowledge producers. Science is often wrongly portrayed as an elitist activity accessible only to those endowed with large grants and sophisticated laboratories and wearing white coats. Good science can be practiced by anyone with a sharp mind, even without any grants and laboratories. Science should become a democratic, grassroots activity, accessible to all. As I said in the title, young and old, rich and poor, amateur and professional, student and teacher. Why only teacher? Even students. As I said, I have written a book which describes 15 such simple experiments anybody can do, and I describe them in great detail, and they're all in this book, and this book is freely available to any of you. You can go to this website, you can write this down or take a photo of this slide. You can go to this website and you can access the book for free. There are 15 chapters with 15 experiments, and there is a 16th chapter saying why low cost experiments are important. Now I wrote these, I used to write them regularly, and they were published in a journal called Resonance, and then they were made into a book. When it was over, I was very unhappy, because the destination is not important. I wrote the book, it's over, it's published, what do I do now? I wanted the journey to continue, I wanted a circular train. So what I have done is, since the book is over, I have started writing more such chapters, more articles regularly, and publishing them in a column which was mentioned in the introduction called More Fun Than Fun. This is my column called More Fun Than Fun, and I have just illustrated 6 examples here. So far I have written 40 chapters, all of them simple experiments, simple facts, and this is also freely available at this site. You can choose any that you like and read about it. Okay, so depending on the time we can have questions. I don't know how much time we have.
Thank you very much, sir. I'm sure we can have questions, so we can start for the guys at the back. Sir.
How did you get ideas about
Ideas about?
About experiments.
How do you get ideas? By being curious. It depends on the context. So you go out, if you go out and simply look at leaves on plants. Leaves have different shapes, no? Are all leaves have the same shape? They have different shapes. Why? Different flowers have different colors. Why? Different flowers have different types of petals, different numbers of petals. Why? Some trees grow short, some trees grow tall. Why? There is an endless number of questions. You must try to think about them and see how you can try to understand. Anything that you look at, you can ask questions. So ideas are not a problem. Then you have to think about it. So for example, let me take the first one I said, shapes of leaves. How will you begin? You will say, okay, how many different types of shapes are there? Let me go and look at it. Let me find out, let me classify how many kinds of leaves there are. And then try to imagine what are the advantages and disadvantages of different shapes. Okay? And then start making a guess. You can say, if a leaf is very broad, then it is easy to absorb light and produce a lot of photosynthesis. That's an advantage. What is the disadvantage? Good food for insects. They can come and eat all of it. You begin. So you have to ask questions first. Anything that you look at, immediately, it should not happen that something does not raise a question in your mind. Okay? So if I look at you, I can ask a simple question. Why is it that you are sitting in this pattern? Did someone tell you this is your place, you sit here, you sit here, you sit here? Probably not. Why did you sit like this? What made you sit in this? What provided this configuration? So this is a question probably I'll never be able to answer, but that doesn't matter. You should train yourself to raise questions in mind. Anything that you ask, you raise questions. In Andhra, people eat very spicy food, right? Why? Anything in this world, you can ask a question. Why or how? Why do people eat spicy food in Andhra compared to Karnataka? The answer is not important. The ability to ask questions. Then start thinking. Now, for this question, if you want to start thinking, you can look more broadly. Let's not restrict to Andhra. You have to get more information to begin. So let's not say Andhra and Karnataka. Let's look at the whole world. Then you will find that in the tropics, people eat spicy food. Near the poles, people eat non-spicy food. Now you can begin to think, there may be a reason. You may be able to explain. So this is how you have to begin. Train yourself not to be satisfied. Somebody tells you something, don't be satisfied. Dissatisfaction is the main thing that you need. Don't be satisfied. Ask a question. Unfortunately, I know that in many schools, students don't ask questions. That is the wrong thing to do. If that happens in your room, outside the classroom, you can ask questions. No, nobody will stop you. So learn to ask questions. Never be satisfied with anything else. But in fact, children who are never satisfied, keep on asking questions, are considered very bad. But they are the best students. They are the ones who will become scientists.
Sir, why did you do many experiments on ethology and
Why did I do? Because I love animals. I love animal behavior. Passion. I love it. Do what you like. There are many things you can do, but you should also say, I don't like that. Fine. I don't like chemistry, or I don't like biology. I don't like maths. Doesn't matter. You should do what you like. That's why my column is called More Fun Than Fun. You should have fun. Then you will put your best. You may have to learn all subjects because of some background that you need. But your own concentration can be on what you love. So find out what you like. You may not like to be interested in understanding why leaves have different shapes. You may be interested in understanding why insects have 6 legs. Fine. Follow your heart. Follow your passion. Follow your interest. Never be satisfied. Any other?
So I have a question that.
Mike sir illalla.
You speak up. I can hear you. Speak up loudly.
So I have interest in this field, and I want to know that. Can I mean, I just don't get it now.
That's okay.
So my parents say that this field wouldn't benefit me much, but I really am interested in studying about ants, insects. So I just want to know that if this field would, like benefit me.
There are many ways of dealing with this problem. It's true. There are many reasons why many of us cannot follow what is closest to our heart. You are not unusual in that. Many reasons. One is parents tell you do this, you become a doctor, study for medicine. That is one. Teachers tell you don't do this, do that. Various reasons. I don't have money, I don't have time. Hundreds of reasons. Many of us cannot follow what we really like to do as our main profession. That is why science should be a hobby. Everybody is entitled to have a hobby. Your hobby may be swimming, cycling, running, stamp collecting, whatever. Have a hobby. Nobody is occupied 24 hours. Make science your hobby. In fact, the best scientists make science their hobby. So have it as a hobby. You do whatever you satisfy your grandmother and become a doctor, no problem. Treat your patients well, but you have some spare time. Go and look at ants. Nothing prevents you. The reason why people think it is not possible to pursue science as a hobby is because they think science is a very sophisticated thing, needs a big laboratory, lots of time, lots of money. This is the problem. The moment science becomes like this, anybody can do science. You can always find an hour, half an hour, one weekend, go out and look at ants. Nobody prevents you. So don't worry that cannot be, at this age you can't say, how will I get Nobel Prize on ants? No, you don't worry about Nobel Prize. If you are passionate, if you are interested, you can always pursue your questions as a hobby. Make science your hobby. In fact, many scientists are scientists by profession, but they do one kind of research. But they do something else as a hobby, and that gives them much more pleasure, and often that gives them much more awards and rewards than what they do as a profession. I know many scientists who pursue one kind of science as a profession for which they are paid, but a completely different kind of science as a hobby for which they are not paid. And most of them tell me that I enjoy this much more. So it's never a problem. There is no dearth of questions, and if it does not require anything else, make it your hobby.
ನೀವು ಕನ್ನಡದಲ್ಲೂ ಕೇಳ್ಬೋದು questions.
If you are not going to ask me questions, I am going to ask you. Yes, you had a question? Yeah, please.
So, like,
why do honeycomb is made up of hexagonal rings and
Excellent.
why not any other shape?
Excellent. So it's a very good question. Now, I can give you the answer, but you will hate me. So then what are you going to do? Try to find the answer. See, this is what we have to learn. I can tell you the answer, the end of story. Whereas, simply as a hobby, I can assure you that you can spend the next several years trying to understand this question. Because the final answer is not known. There are many answers, but nobody really knows. Find out. How will you begin? See, the best thing, most important, see, half the battle is won. You are already 50% scientist. You asked a brilliant question. The remaining 50% is trying to answer that question. Don't answer that question usually by blindly going and reading. Then you will only get the answer. Don't read. Try to think. Yeah, exactly. That's what I said. So try to ask that question. Answer that. Think about it. Now, how will you begin? I'll give you some hints. It is, you're saying, why is it hexagonal? You must ask, what else could it be? Think like. Then you say, okay, why not a circle? Now you compare hexagon and circle. What are the advantages? What are the disadvantages? And see whether you can come up with any argument why hexagon is better than circle. And so that's how you proceed. Okay? So, wonderful questions, but I hope nobody will spoil you by giving you the answer. Find the answer. Not by reading, by thinking. If somebody asks me not an important question, I will probably give the answer. But such brilliant questions, I will never be cruel to give the answer. So you have to find the answer. It's a very good question. Still being researched. And this question has been asked for thousands of years. Thousands of people have tried to answer that question. We don't know the final answer. Maybe you will find it. So don't be satisfied with the answer. Yes.
Sir, why do honeybees prefer only nectar or sugar syrup but not spicy things?
Not?
Spicy things.
Spicy things. If you don't give them anything else, they'll probably take somewhat spicy food. But you see, honeybees use nectar as a source of carbohydrate and pollen as a source of protein. They need carbohydrate and protein for bringing up their larvae. That is the main thing they need. Now, within that there may be a little bit of spice they may tolerate, but they are looking for sugar because that is what they need, a source of energy. See, when we say we like spice, we don't eat only chili powder, right? You just add a little bit of chili to make your food palatable. You're not getting proteins and carbohydrates from chili powder, right? So that's only an add-on for your taste. Now why because you are fussy. If the food is bland, you don't eat. But honeybees are not collecting food for themselves. They have to feed all the larvae. So they have to feed whether it is palatable or not. But if there are two kinds of honey, they will choose the one that they like. But again, don't be satisfied with my answer. Try to find out. Give some spice to honeybees, see whether they will like it. Maybe one kind of spice they like, another kind of spice they will not like. Or if they take the spicy food, they may die. See, no answer is final. You can try to find out. And if it's difficult to work with honeybees, try some other insect. Ants are easier to work with. Try to give some spicy food to ants and see whether they'll take. Maybe they won't take. Then you starve them. If they're very hungry, will they take? Maybe they'll take. If they take, maybe they'll die, maybe they'll not die. You can do this experiment at home. So no answer is final. Investigate.
Thank you, sir.
There is one question on the chat. You
Yes.
want to take that one? It says, why does honey never expire? What is the reason for it?
Yes. Okay, so this is a question I will answer because not easy for you to find out yourself. The honeybees add many kinds of antibiotics to the nectar so that it doesn't go bad. That's one reason. The second reason is they concentrate it so much that it is difficult for bacteria to grow in it. But don't be satisfied with the answer. Ask yourself, why is it difficult for bacteria to grow on concentrated honey? And why is it easy for them to grow on dilute honey? I am telling you that one reason why honey usually does not get spoiled is because it is very concentrated sugar solution. But you must not be satisfied with the answer. You must think, why is it difficult for bacteria to grow on concentrated sugar and easy to grow on dilute sugar? That question I will not answer. You have to answer that yourself. So the two main reasons, one is it is very concentrated sugar solution, the other is that antibiotics are added to it already. Both of these are important. Antibiotics part is okay, but the concentrated part leads to another question. Every answer leads to another question. That is why science is never ending. That is why it is endless joy.
I think we can take one more question.
Somebody's
Sorry, I
standing
just
up already.
promised.
Okay, sure, anybody.
So like like all the bees follow the queen bee, no sir?
Yes.
So what happens if the queen bee dies? Like what happens to the other worker bees and everything?
In the case of honeybees, workers cannot become queens. Queens cannot become workers because they are born as queens and workers. And therefore, if the queen dies, the whole colony dies. Except special situations. Workers cannot become queens, but they can rear a new queen. Now, how do you rear a queen? To rear a queen, you have to give that larva, when it is very young, a very special kind of food. This special kind of food is called royal jelly. Now, if the honeybee queen dies, and if there is a young larva, then they can give it royal jelly and get a new queen. But if there are no young larvae, then the whole colony dies. So workers cannot become queens, but they can rear a new queen provided some very young larvae or some eggs which have not yet hatched are available when the queen dies. This is called, it's got a nice name, it's called emergency queen rearing. But they can only do emergency queen rearing if there are eggs or very young larvae. But the honeybees will never accept a foreign queen. They will die, but they will not accept a foreign queen. Their own queen, or they will make a new queen from the eggs in their own colony. I have made you ask many questions. Why should they not accept a foreign queen? Why only young larvae? What is royal jelly? Why does it make a queen? How does royal jelly make somebody a queen? There are endless questions. You must pursue these. An answer is not an end. An answer is the beginning of many new questions. You can pursue all of these questions. That is why I said science is endless. And all of you can do this. What prevents any of you from asking this question? Why is honeycomb hexagonal? Do you need money to ask that question? You don't need. All of you can become scientists. This is what I want to tell you.
Sir, why can bulls visible red color only, sir?
Sorry?
Bulls can visible red color only, not other colors.
Red color, bees cannot see red.
He
sir,
said
bulls.
that he thinks that bulls can see red while they can't see others. That's what he said.
They can see only red.
Like that, sir. They can visible the red color, no, sir.
You remember what I said about black and white television? It can appear as a shade of grey, isn't it? Without actually having color vision. But you can do an experiment. Bulls are not very hard to come by. Do an experiment. It's not difficult. You see, you don't even have to do the experiment. Think of an experiment. Come up with an experiment which somebody can do, but you have to come up with an experiment. The three experiments I described, even if any of you have written down the exact experiment that needs to be done, that is more than 50%. So it may not be possible, but think of an experiment and then think of what is wrong with your experiment. Why it is not enough to show that pine cones make the difference. Why it is necessary to also put scented cardboards. Why it is necessary to descent the pine cones. Why is it necessary to cut off the antennae. So all of this can be done as a thinking process, even if you can't actually do the experiment. What experiment will you do to prove whether the bull has no color vision and is seeing red as a shade of grey, or it actually has color vision but only one kind of color cells which will see red and not others? What experiment will you do? You can think about this.
So there are actually several other questions. I will put out just one of them.
Okay.
He says that even plant species flowers must have disease, and the bees that come and collect it might also spread disease.
Absolutely. Not only they spread pollen from one flower to the other and bring about cross-pollination, but they also spread disease. Yes, that's true. But life is a trade-off. If the flower says, no, I don't want to have any bees because it may bring disease, then it will not get pollinated. So there is risk, you have to take some risk in everything. So flowers have survived for millions of years in spite of using bees to transfer pollen from one flower to the other. But there is just definitely risk of disease. We have to trade off. There is a risk of disease. My coming here and speaking to you for 1 hour without my mask is a risk, but I have taken it.
So suddenly there's actually a spate of questions, but it appears that you have some other appointments to keep.
Yes.
So I'm going to...
One option is that students are free to send me their questions by email.
I was just going to say that
yes.
I'm sure Professor Gadagkar will be happy to answer questions by email.
Yes.
We can have Professor Gadagkar's email ID put out.
It was on the first slide. In case you have not copied it, you can get it from them. Yes, feel free to ask me questions.
Also the online
But
audience.
it may be possible that I will say this is such a brilliant question, I will not answer it, find out for yourself. That also I might say.
Yeah, so also to the online audience, you can collect the email ID. We will put it out at some point. Thank you very much, Professor Gadagkar. I'm sure all of you enjoyed this lecture.
Okay. Thank you, sir. Thanks for the wonderful talk. And now I request Professor Shiba Vasu to give a small present on behalf of CNR Hall of Science and Education Technology Unit.
Oh, thank you so much. Thank you very much. Yeah. Oh, one more. Thank you. Thank you.
Due to time is limited, and if you have any, because we have series of questions over there, from the online participants, and please, I will share all the questions to the professor. And if you have any questions, some more, you can directly send an email to professor. And now it's time for vote of thanks. Once again, on behalf of CNR Hall of Science and Education Technology Unit, we express our thanks to Professor Raghavendra Gadagkar for such an interesting lecture. And our thanks to Professor Shiba Vasu for convening the program and introducing the speaker. I thank Bharat Ratna Professor C.N.R. Rao and Dr. Mrs. Indumati Rao for their vision, continued guidance and commitment to popularizing science in nationwide. And you can see Mrs. Rao is here. And we thank the President of JNCASR, Professor G.U. Kulkarni, for extending his support and facilities for this program. And we appreciate the participation of JNV students and teachers from various colleges across India from this today's event, both online and offline. My special thanks to Dr. Gopal Krishna, Deputy Commissioner JNV in charge, and from Vidyan Jyoti program. He coordinated all the colleges and across India. And we thank the supporting staff and administration for the JNCASR for providing the infrastructure, facilities, and administrative support for this program. Finally, I thank Mr. A.N. Jayachandra and Praveen, Ms. Sukanya, and Mr. Dilip and Munegoda for their technical assistance. And thank you very much, and thank you once again all.